Research out today in the journal Cell shows that a specific change in the SARS-CoV-2 coronavirus virus genome, previously associated with increased viral transmission and the spread of COVID-19, is more infectious in cell culture. The variant in question, D614G, makes a small but effective change in the virus spike protein, which the virus uses to enter human cells. Bette Korber, a theoretical biologist at Los Alamos National Laboratory and lead author of the study, noted, the D614G variant first came to our attention in early April, as we had observed a strikingly repetitive pattern all over the world, even when local epidemics has many cases of the original form circulating. Soon after the D614G variant was introduced into a region, it became the prevalent form. Geographic information from samples from the GISAID COVID-19 viral sequence database enabled tracking of this HIG. This occurred at every geographic level: country, subcountry, county, and city. Two independent lines of experimental evidence that support these initial results are included in today's paper. These additional experiments, led by Professor Erica Ollmann Saphire, Ph.D., at the Jolla Institute, and by Professor David Montefiori, Ph.D., at Duke University, showed that the D614G change increases the virus infectivity in the laboratory. These new experiments, as well as more extensive sequence and clinical data and improved statistical models, are presented in the Cell paper. More in vivo work remains to be done to determine the full implications of the change. The SARS-CoV-2 virus has a low mutation rate overall, much lower than the viruses that cause influenza and HIV-AIDS. The D614G variant appears as part of a set of 4 linked mutations that appear to have arisen once and then moved together around the world as a consistent set of variations. "It's remarkable to me," commented Will Fischer of Los Alamos, an author on the study, both that this increase in infectivity was detected by careful observation of sequence data alone, and that our experimental colleagues could confirm it with live virus in such a short time. Fortunately, their advertisement Korber noted, these findings suggest that the newer form of the virus may be even more readily transmitted than the original form. Whether or not that conclusion is ultimately confirmed, it highlights the value of what were already good ideas, to wear masks and to maintain social. That work also included observations of COVID-19 patients from Sheffield that suggested an association of the D614G variant with higher viral loads in the upper respiratory tract. It is possible to tracking SARS-CoV-2 evolution globally because researchers worldwide are rapidly making their viral sequence data available through the GISAID viral sequence database, Korber said. Currently tens of thousands of sequences are available through this project, and this enabled Korber and the research team to identify the emergence of the D614G variant. GISAID was established to encourage collaboration among influenza researchers, but early in the epidemic the Consortium established the SARS-CoV-2 database, which soon became the de facto standard for sharing outbreak sequences among researchers worldwide. The study, tracking changes in SARS-CoV-2 Spike, evidence that D614G increases infectivity of the COVID-19 virus was supported by the Medical Research Council, MRC, part of UK Research and Innovation, UKRI, the National Institute of Health Research, NIHR, Genome Research Limited, operating as their I120070010000, and the Los Alamos Laboratory Directed Research and Development Program. Additional study authors included S. Nakaran, H. Yoon, J. Thyler, W. Abfalterer, N. Hengartner, E. Georgi, T. Bhattacharya, B. Foley, K. M. Hasty, M. D. Parker, D. G. Partridge, C. M. Evans, T. M. Freeman, T. I. Da Silva, C. McDaniel, L. G. Perez, H. Tang, A. Moonwalker, S. P. Whelan, C. C. La Branch. Thanks for watching. Do you like my video? Then show me your love by pressing like button. Please subscribe my channel and hit bell icon for new science inventions in future.